Hello and welcome to the next lecture in this unit, that is the sources of contamination in food. When we speak about food, and in the last lecture as we saw that food is basically a combination of various chemicals, it gives a good opportunity for the microorganisms to settle on the food and multiply. This most of the times is not desirable by the humans who use these foods for their consumption. So microorganisms, they get into food from both natural sources and from external sources to which the food comes into contact from the time of production until the time of consumption. Right from the point where it is prepared to the time where it is consumed, there is a whole lot of a chance, huge probability that the food will be contaminated with microorganisms. Natural sources for food of animal origin means, as I said, there is livestock, there is feedstock. Livestock is animals being used as food, meat, chicken, poultry, etc. Animal sources itself, they become what you can say, source of contamination, like hair, skin, feathers, gastrointestinal tract of that organism, urogenital tract, respiratory tract, milk ducts, that is the teats in udders of milk animals. This we will study in detail in milk microbiology in the last unit. But the animal itself becomes a source. The natural flora which exists in ecological balance with their hosts and their types and the levels, they vary greatly with the types of plants, animals, as well as their geographical locations and environmental conditions where the organism is living, from where it is being derived as a source of food, its habitat, the food which it eats, and how it is handled, where it is kept. All this plays a very important role in the addition of microorganisms in food when it comes to the livestock. Besides the natural microorganisms which these animals harbor, the foods can also be contaminated with different other types of microorganisms which come from other sources from where the the animal is handled, like the air, soil, sewage, then the utensils, humans who are handling these animals or the foodstuffs, food ingredients, packaging, the packaging environment, the packaging containers, insects, etc., vermins which are there in and around the environment or the place where this food is being manufactured. Microbial types and their levels from these sources getting into foods, they vary widely, and they depend mainly on the degree of sanitation which is used during the handling of foods. Whenever we go for outside food, what we desire is the food should be of good quality, and what makes this good quality stuff is how it is being prepared, under what conditions it is being manufactured, right from the person who is manufacturing to the utensils which are being used and the environment in which it is uh, being produced. Remember, when a food is being prepared in a closed environment for years together, it becomes a very good environment for the microorganisms to adapt and thrive even without being noticed. So the types and their levels, that is, they vary greatly, and they depend, it totally depends upon the sanitation, how clean that food kitchen or the environment where the equipments, the person who is handling, how well healthy, good they are. This is one of the important aspect in food quality. So microorganisms, as I just said, they can be added from different sources. Huge variety of sources are responsible for addition of microorganisms in food. Usually, while listening to this lecture, don't have an idea or don't get idea that whatever the microorganisms they are getting added, they are bad. It is not so. Most of the times, natural flora which is usually harmless may get added, and most of the times if the food is cooked well, the microorganisms, they get killed, they are removed. But the compromise of heating, that is the temperature used for heating, the compromise is between the nutrition and the temperature used. There is a very subtle difference uh, between this. So that is an art. That is where the food cooking becomes an art. So microorganisms, they can be added from different sources, plants, that is fruits and vegetables, which we use, the livestock and the feedstock, which I was talking about, feedstock now, the agricultural sources, the inside tissue of the foods from plants, they are sterile. That is, if you take the inner part of a fruit, it is sterile. Only if the vegetable or the fruit is porous, like radish, onion, cabbage, these are porous in nature and they may have microorganisms within. But most of the plants, they produce antimicrobial metabolites which can inhibit the microorganisms, like garlic is there, onion is there, they have an inner potential of synthesizing antimicrobial agents. So that is a separate area of study, but microorganisms, they are present when it is coming from the agriculture, because it grows in soil. Fruits and vegetables, they harbor huge variety of microorganisms on their surface, as I said, because they are coming from soil. Their types and their levels, that is which type of microorganism and what is the level of contamination, it depends and varies greatly with respect to the soil condition. If it is a good fertile soil, try to understand, then there will be a lot variety of microorganisms, type of fertilizer which is used, the water quality amount which is used, and the air environment, that also determines the type and quantity and quality of microorganisms in your food. So microorganisms which are present in plants, they usually are molds, yeasts, lactic acid bacteria, pseudomonas, alkali genes, micrococcus, irvania, bacillus, clostridium, enterobacter. All these bacillus it is one of the major components of soil, so it is bound to be there. Pathogens like enteric types can be present, means those which are present in intestine of animals, they can be present only if it is contaminated with defecated matter or the sewage. If sewage is being added into agriculture, then there is water which is being added to agricultural soil. If it contains sewage, then there is a lot of chance, huge chance, that enterobacteriaceae members can be present in that. The factors which affect the number of microorganisms in plant, they include disease of the plant, diseases which are various types of disease which cause, if a fruit is getting rotten, then the microorganisms, viruses, bacteria, yeasts, molds can be present in that foodstuff. Long delay between harvesting and treatment, that is washing, cleaning or disposal into the markets, selling off. If there is a large time between harvesting and selling, then, or unhygienic conditions if they are present in the storehouse, then there will be increase in the number of microorganisms. Unfavorable storage and transport before processing and improper storage after processing is also responsible for microorganisms in plant stuffs. How to reduce the microorganisms in plants, I am saying, because it is fruits and vegetables, they are major sources of food, just like the animal meat, chicken, poultry, eggs, etc., milk, etc. So reducing the microorganisms in foods from plant involves use of treated sewage for fertilizers. Clean water is a rare commodity. If the wells are dried off, if the canals are not providing water, then most of the time what happens, these sewage waters, they are drifted or they are diverted into the agriculture. But if it is treated sewage, then there will be no harm. Damage reduction during harvesting, when it is post harvesting conditions and facilities, if they are properly maintained, like the cleanliness, hygiene, temperature, moisture, etc., if they are regulated, then there will be no contamination. Quick washing with good quality water to remove soil and dirt. Whenever you bring fruits and vegetables into your house, you should take care that it should be immediately washed with clean water and dried off. Same thing applies to storage conditions post harvesting. And if the processed agriculture plants, vegetables, they are kept in low temperature, then there will be less microorganisms. So we have cold storages for this. Now from the livestock, like animals, birds, fish and shellfish, that is the seafoods which are being used as food, they carry many types of local microorganisms or indigenous microorganisms in the gastrointestinal tract, respiratory tract, urogenital tract, teat canals, in the udders, as well as skins, hooves, hairs and feathers of the animals, birds, fish, shellfish, etc. All their body parts which are exposed to either air or soil and water. You see, if it is seafood, it will be exposed to water. If it is animal like chicken, that is sheep and lambs, etc., they will be exposed to soil. So this brings in microorganisms. Number of these microorganisms depends on the specific organ. If it is inside the intestine, then 10 raised to 10 microorganisms per gram of meat. Try to understand the intestine. These are cleaned. There is a food processing procedure, which a protocol which has to be followed. If it is not proper, then there is a huge chance that the intestinal organism of the animal will be added into the food. There will be spore forming also. Same with the respiratory tract, same with the of the animal or the fish or the birds, that I am saying. Same with the urogenital tract, same with the respiratory tract, other parts, skin, hairs are present. All these contribute to huge number of microorganisms in the food. Many animals, they act as carriers. They harbor pathogens like staphylococcus aureus, which is responsible for infections in the wounds, E. coli, which is an opportunistic pathogen, campylobacter, listeria, without showing any symptoms. They can be very dangerous to the consumer. Laying birds, that is birds which are having eggs inside and they are about to lay the eggs, they carry streptococcus enteritidis in their ovaries, and this is a pathogenic condition. So fish and shellfish, that is coming from the sea, they carry normal flora in their scales, skin and digestive tracts. All they harbor microorganisms. They can also carry various types of pathogenic bacteria. Parahemolyticus is one of the organism which is present in the digestive tracts of the sea animals. Many spoilage and pathogenic microorganisms. You have to understand two different things. Spoilage of the food is also non-desirable, and if pathogens develop, they will go inside our body. They are also not desirable. They can get into the foods of animal origin like milk, egg, meat, fishery products during production and processing. Milk can be contaminated with fecal material or from the udder surface. This we will study in detail in the last unit. There is a separate unit called as milk microbiology, but you should understand that the milkman, the skin, the udder, the teats, the organ of the milk production of the animal is equally responsible for addition of microorganisms in the food. That is, milk is one of the most universal food products which humans consume. Meat similarly can be contaminated with intestinal contents during slaughtering, during that is cutting and preparation of meat. The intestinal organisms can come and get added. Meat from food animals, that is the livestock and the birds or the seafoods, can be contaminated with different types of spoilage and pathogenic microorganisms from various organs or the body parts like skin, hair, feathers, etc. And predominantly Staphylococcus aureus, Micrococcus, Propionibacterium, Corynebacterium, molds, pathogenic yeasts like Candida, etc. They can be present and they can be added in the food. Partially or most of it they are destroyed if the food is heated, boiled, fried, etc. But in many cases, if neglected, if the processing is not good, then they may be added in the food. How to reduce the contamination from animal sources? There are good practicing techniques, good housing and supply of uncontaminated feed and water. If you give good food and good water to the animals which you are cultivating for food processing, like chickens, poultry, or the sheep, then they have to be provided with healthy diet, healthy food and good water. Testing of these animals and birds for pathogens and culling carriers, that is during their slaughtering, the amount of pathogen risk goes on increasing if they are already contaminated, so they should be properly checked and tested using good quality of water for washing the carcasses, means the dead. Once it is slaughtered, it is a carcass, so you have to clean it. There will lot of bloodshed, and various other organs will spill various different types of toxins and enzymes and body chemicals, hormones, etc. They have to be cleaned and washed off. Hair removal, removal of contaminated parts, proper cleaning of the udder before milking in the case of milk products. These are some of the good manufacturing practices. If it is milk, it should be cooled immediately after milking, and uh, after milking it should be heated, immediately cooled, and then processed. Collection eggs should be done immediately after laying. If after laying it is remaining there, it will go towards hatching. There will be chicks produced, and the amniotic fluid which is present inside it undergoes lot of changes, and improper timing may result into contamination. Fish and seafoods, marine products, they should be harvested from unpolluted water and stored properly. These are some of the practices. Now next is air as a source of contamination. Microorganisms, they are present in dust and moisture droplets in the air. We have seen these things in our last semester in air microbiology, how microorganisms are what you can say universally present in air. Even though, remember, air is not a good medium for growth medium for microorganisms, but when it comes and settle down on the food, they are omnipresent. When they settle down on the food, they, via dust and moisture droplets in the air, they allow themselves to grow and form huge colonies. Microorganisms in the air, they are transient and variable, they are dynamic, and they can settle on any and every type of food. Level of microorganisms in the air, they can be easily controlled by adjusting the humidity. More dry the environment is, less will be the organisms which will settle, but more will be the dust. It is a fine balance which is an art, and it has to be controlled. Level of dust particles minimum 0. Temperature should be very ambient. Air velocity and resistant to drying. Air velocity, if it is a turbulent air there, you don't have fans in the kitchen. Why? Think about it. It is not always due to the fire, but it is also due to the circulation of microorganisms. And drying of the food should be avoided by controlling the humidity. Dry air with low dust content and higher temperature with low microbial levels can be achieved using these parameters. Some of the examples of microorganisms in air are spores of Bacillus species, Clostridium species, molds and yeasts, cells of gram-positive bacteria like Micrococcus, Sarcina, etc. Now, if the surroundings it contains pathogens like the poultry, animals, or if there is a sewage treatment plant, different types of bacteria which include the pathogens also, they can be transmitted via air in and around which environment your food processing is. As an example, let me tell you, there is a kitchen, and if you open the window, there is a huge sewage treatment plant, or there is a big nala. Now what will happen over there is the air will be responsible for transporting the droplets of sewage into the food. So conditions have to be controlled. Control measures for this is removing potential sources, air filtration, reducing the humidity, installing UV lamps. These are some of the control measures for controlling the microorganisms in the air which can be added into the food. Next important source of contamination in food is soil. As you know very well, it is a main part of agriculture. Soil is used in agriculture to produce and raise the plants, even animals, because animals, they have to be fed with the plant materials. The birds, they are given grains, etc., which harbors or transfers and transports variety of microorganisms. Microorganisms, they can multiply in soil, and they can be in billions. 1 gram of soil minimum, it contains 10 raised to 8 to 10 raised to 12 microorganisms per gram. So it is a huge number. Microorganisms which are usually found in soil and which can become a part of the food are molds, yeasts, enterococcus, bacillus, clostridium, enterobacter, pseudomonas, proteus, micrococcus, and various other parasites. Soil contaminated with fecal materials can be added into the soil. Usually in developing countries we have open defecation. Try to understand this as applied microbiology concept which we saw. It is an applied thing that we should not defecate in open. We have government schemes where government allows you to construct a laboratory latrine and bathrooms and encourages you to use those and discourages you to openly defecate. Why? Because soil gets contaminated with fecal matter and source, it becomes a source of enteric pathogen and viruses. Sewage, as I just said, can also be a source of contamination in food. Milk is also food. Plants and vegetable sources like vegetables and fruits are also food. Meat, egg, milk, these are also food. All can be added with sewage because every part needs water, and for washing, cleaning, dilution, cooking. If this water itself is not clean, if it is sewage coming from a sewage source, how this is possible? We saw it in water microbiology and sewage microbiology. Sewage can contaminate clean water, and clean water which is used for food processing can get contaminated. So sewage can contaminate basically crops with microorganisms when it is used as a fertilizer. That is enteropathogenic bacteria, viruses, parasites, everything can be added. Sewage should be treated before using it. As we see, you see, we have discussed the sludge being used as a biofertilizer. The point comes here. Sewage as a contaminant in agriculture, crops, foods, the link if you can establish. We have said the disposal of sewage, it is a very good biofertilizer, very good. But if it is untreated sludge, if it harbors pathogen, then it can add pathogens into the agriculture. So that too also may be allowed, but later on effective treatment, post processing, cleaning, effective washing of food and food parts should be there after the harvesting is done. Having said that, water itself can become as a source of contamination. The next source of contamination is water. Contamination of foods with pathogenic bacteria, viruses, parasites, they have been recorded time and again. Wastewater, it is recycled and used for irrigation, so that recycled water can become source of contamination if it is not treated properly, if it is not chlorinated, etc. Potable water which we use from good sources, well, let us say groundwater or canals, they can also contain bacteria capable of causing food spoilage. If not pathogenic, if not harm to the health, they can cause spoilage of food. There are two aspects which you should understand: spoilage of food and subsequently the pathogenic, the health hazard. Second aspect is health hazard. So improperly treated water, it can contain pathogenic microorganisms as well as spoilage causing microorganisms. So water which is used in food processing, it should be of higher microbial quality. Water used in food processing should be of higher microbial quality than that of a potable water. This is one of the very important point. You cannot use potable water all the times. We use treated water or hard water, or let us say well water, groundwater, but the number of microorganisms which is present plays a very significant role. The coming to sources, human itself, the next source I can say, human itself is a big source of contamination. Our lifestyle, our habits, most of them, they have very odd habits of picking the nose, putting fingers in their ears, always touching the hair, combing the hair. These activities can become a huge source of contamination. Means between the production of a food and consumption, food comes in contact with different people who are handling foods. As a food is getting prepared, there is a person involved in it, transporter, person involved in it, consuming, huge lot, a chain of humans, they are present in between the source and the consumer. Like the people working in farms, people working in processing plants, people working in restaurants, the waiters, the cook, etc., the cleaner, etc., people in retail stores, in homes. Humans, they have been a source of pathogenic microorganisms food always. Healthy practices, you can, the best example is of pani puri. It is one of the worst examples of humans as a source of contamination. If you just go on seeing the production to the consumption of pani puri, there is no heat involved in it, then there is storage is large, the water which is used is unclean, the nails, the hygienic conditions, etc., etc., it's horrible. So humans itself can become a source of contamination. Human contribution in contamination is mostly due to unclean, improperly cleaned hands, lack of aesthetic sense and personal hygiene. Aesthetics, what we are wearing, how we are behaving, what are the activities which are required for a particular time, food preparing, how your body language should be, all these things plays a very important role. Dirty clothes, dirty hair, presence of minor cuts and infection on the hands and face. Then most of the time we have, if there is a cut on the face or cut on the hand, we have a habit of touching it again and again because it itches. And if the same person is there preparing the food, he will give his microflora to you through the food. Remember, this is after processing. The waiter who is there, the cook who is there, they can become large contributors of contamination. And if that person is infected with influenza, throat infection, hepatitis A, other type of diseases, then he is going to contribute it to you. So pathogens such as the wound infection, Staphylococcus aureus, which is a pus forming bacteria in humans, Salmonella, Shigella, the enteric bacteria, pathogenic E. coli, hepatitis A, they can be easily introduced into the food from human sources. How to control it? Proper training of personnel and personal hygiene, personnels, people who are handling the food. There is a course, BSc in food science and technology, BSc in hoteling industry, a management degree, hoteling, all these they teach these things. It is not that someone fine day I think of putting a hotel and you just go and put a hotel and then serve people, you are the biggest contributor of disease to the society. It doesn't work that way. Regular checking of the health of the persons who are involved in preparing the food, maintaining efficient sanitary and aesthetic standards, these are very important for a good manufacturing practice. Now food ingredients itself can also become a source of contamination. Food ingredients, food which is being used, all this plus the next source is the food ingredients like the spices, basically which is added, it is an ingredient. I am not saying spices is a source of contamination. I am trying to tell you food ingredients. Spice is there, many chemicals are there, carbohydrates, proteins and lipids are there. Okay, now we add spices. If the spices are not prepared in hygienic conditions, okay, the ingredients which we mix and prepare a basic raw material before cooking, composition, we mix lot of things, we cut vegetables, we cut fruits, then we add salt, pepper, sugar. Preparation involves food ingredients. Many ingredients, they can be source of spoilage and pathogenic microorganisms. If not pathogenic, then spoilage. Remember that. Spices, they have high content of spores. Spores, they can withstand high temperature, and later on when the food is cooled down, they can become a source of contamination. So these ingredients, they should be prepared under good sanitary conditions. Setting up of microbial standards specification for the ingredients is given by FSSAI, that it is the Food Authority of India. We will see that afterwards. There is a regulatory body for all these things. A government body is there and they set up the standards. Under these standards, food preparation should be there, especially when the food is used to serve the community at large, when many people are consuming that. Next source of contamination is the equipment itself. Equipments, utensils, cans, pouches, they all can be sources of contamination. Many microorganisms, they can get into food from equipments, and these organisms, they include Salmonella, Listeria, enteric bacteria, Micrococcus, Escherichia, Pseudomonas, Lactobacillus, Leuconostoc, Clostridia, Bacillus, yeast and molds. See, these are repeating again and again. These organisms, most of them, they are opportunistic pathogens. At source, they cannot be called as very harmful, but when they go inside and their load increases, when they multiply, they can become a great source of contamination. Proper cleaning and sanitation of equipments should be emphasized to reduce the number of microorganisms in food. Lastly, I come to a point where I can say some miscellaneous sources of contaminations are also responsible for the food contamination, like the packaging and wrapping materials, the containers which are used, then the flies and insects which are present in and around mosquitoes. Flies, maggots, they are responsible for maggots. Vermins, small insects which are usually present in the rice or the floors. Birds in and around outside, they are always flying here and there. You can inside the cooking area if they are there, or the storage area, food storage area, then they can also become a source of contamination. The bird droppings which are there, they sit, their bird claws, the bird droppings, the bird pickings, they are great sources of contamination. House pets, usually cats and dogs, sheep and goats, then the cocks and pigeons, these they pets which are used as pets simply not allowed wherever there is a food being prepared. That is a separate long story which is responsible for not only bad health, but over the years a mental condition can also be there. So rodents, rats, lizards, all these inside a production assembly, it should be zero of these things, and proper care should be taken that these should not be there to contaminate the food. So this is all about the sources of contamination in food. I hope you understand this lecture. If you thought that this lecture was very fast, then you have a source at your hands to pause, rewind, re-listen, re-watch, rethink and understand. Above that, for this lecture, if you have any questions, you can put a question in the comment section here, or you can call me, you can contact me in the department anytime and we can discuss your difficulties. But again, I emphasize, listen to this lecture once again with a pen and paper. You can listen to it any number of times. The most important thing which has to be done is to understand. Understanding is simply different from knowing. The first time you listen to this lecture, you will be well informed. You will get information of what is sources, what are the sources of contamination. If you think over it, you will gain some insight of sources of contamination. If you take down the notes and work it out, you will have knowledge about the sources of contamination. When you will practice it in your daily life, then you will be called as a wise person who knows everything about the sources and control of contamination. So I hope I was clear in my lecture. Thank you. We will meet again in the next topic in the next video. Thank you very much.